we are going to have an overview of transcription in both prokaryotic and eukaryotic cells. So transcription is a process in which a DNA strand provides the information for the synthesis of an RNA strand. The enzymes responsible for transcription in both prokaryotic and eukaryotic cells are called DNA-dependent RNA polymerases or simply RNA polymerases. These enzymes are able to incorporate nucleotides one at a time into a strand of RNA whose sequence is complementary to one of the DNA strands, which serves as a template. The first step in the synthesis of an RNA is the association of the polymerase with the DNA template. This brings up a matter of more general interest, namely the specific interactions of two very different macromolecules, proteins and nucleic acids. Just as different proteins have evolved to bind different types of substrates and catalyze different types of reactions, so too have some of them evolved to recognize and bind to specific sequences of nucleotides in a strand of nucleic acid. The site on the DNA to which the RNA polymerase molecule binds prior to initiating transcription is called the promoter. Cellular RNA polymerase are not capable of recognizing promoters on their own but require the help of additional proteins called transcription factors. In addition to providing a binding site for the polymerase, the promoter contains the information that determines which of the two DNA strands is transcribed and the site at which transcription begins. So, chain elongation during transcription. The polymerase covers approximately 35 base pairs of DNA. The transcription bubble, composed of single-stranded or melted DNA, contains about 15 base pairs, and the segment present in a DNA-RNA hybrid includes about 9 base pairs. The enzyme generates overwound or positively supercoiled DNA ahead of itself and underwound, negatively supercoiled DNA behind itself. These conditions are relieved by topoisomerases. Then RNA polymerases in the act of transcription elongation is very important. The downstream DNA lies in a groove within the polymerase, clamped by a pair of jaws formed by the two large subunits of the enzyme. Then the DNA makes a sharp turn in the region of active site so that the upstream DNA extends upward in this, and the nascent RNA exists from the enzyme's active site through a separate channel. Then chain elongation results in an attack by the 3' OH of the nucleotide at the end of the growing strand on the 5' alpha phosphate of the incoming nucleotide triphosphate. The pyrophosphate released is subsequently cleaved, which drives the reaction in the direction of polymerization. The geometry of base pairing between the nucleotides of the template strand and the incoming nucleotide determines which of the 4 possible nucleotide triphosphate is incorporated into the growing RNA chain at each site. So we learned about promoter. Cellular RNA polymerases are not capable of recognizing promoters on their own but require the help of additional proteins called transcription factors. In addition to providing a binding site for the polymerase, the promoter contains the information that determines which of the two DNA strands is transcribed and the site at which transcription begins. So RNA polymerase moves along the template DNA strand towards its 5' end in a 3' to 5' direction. As the polymerase progresses, the DNA is temporarily unwound and the polymerase assembles a complementary strand of RNA that grows from its 5' terminus in a 3' direction. RNA polymerase catalyzes the highly favorable reaction RNA plus NTP gives RNA plus PPI, in which the ribonucleotide triphosphate substrate like NTPs are cleaved into nucleoside monophosphates as they are polymerized into a covalent chain. Reactions leading to the synthesis of nucleic acids and proteins are inherently different from those of intermediary metabolism, whereas some of the reactions leading to the formation of small molecules such as amino acids may be close enough to equilibrium that a considerable reverse reaction can be measured. Those reactions leading to the synthesis of nucleic acids and proteins must occur under conditions in which there is virtually no reverse reaction. This condition is met during transcription with the aid of a second favorable reaction, PPI is equal to 2PI, catalyzed by a different enzyme, the pyrophosphatase. In this case, the pyrophosphatase or PPI produced in the first reaction is hydrolyzed to inorganic phosphate PI. The hydrolysis of pyrophosphate releases a large amount of free energy and makes the nucleotide incorporation reaction essentially irreversible. As the polymerase moves along the DNA template, it is incorporated and incorporates complementary nucleotides into the growing RNA chain. A nucleotide is incorporated into a RNA strand if it is able to form a proper Watson-Crick base pair with the nucleotides in the DNA strand being transcribed. This can be seen and were the incoming adenosine 5' triphosphatase pair with the thymine containing nucleotide of the template. Once the polymerase has moved past a particular stretch of DNA, the DNA double helix reforms. Consequently, the RNA chain does not remain associated with its template as DNA-RNA hybrid except for about 9 nucleotides just behind the site where the polymerase is operating. RNA polymerases are capable of incorporating from about 20 to 50 nucleotides into a growing RNA molecule per second, and many genes in a cell are transcribed simultaneously by 100 or more polymerases. The frequency at which a gene is transcribed is highly regulated and can vary dramatically depending on the given gene and the prevailing conditions. The electron micrograph of this shows molecules of phage DNA with a number of bound RNA polymerase molecules. Then RNA polymerases are capable of forming prodigiously long RNAs. Consequently, the enzyme must remain attached to the DNA over long stretches of template. The enzyme is said to be processor. At the same time, the enzyme must be associated loosely enough that it can move from nucleotide to nucleotide of the template. It is difficult to study certain properties of RNA polymerases such as processing using biochemical methodologies that tend to average out differences between individual protein molecules. So consequently, researchers have developed techniques to follow the activities of single RNA polymerase molecules similar to those used to study individual cytoskeleton motors. Two examples of such studies are very important. In both of these examples, a single RNA polymerase is attached to the surface of a glass cover slip and allowed to transcribe a DNA molecule containing a fluorescent bead covalently linked to one of its ends. The movement of the fluorescent bead can be mentioned and monitored under a fluorescence microscope. As the polymerase transcribes the template, the connecting DNA strand is elongated and the movement of the bead is increased. This type of system allows investigators to study the rate of transcription in an individual polymerase and to determine if the polymerase transcribes the DNA in a steady or discontinuous movement. Then RNA polymerase molecule is attached to the underlying cover slip and allowed to transcribe a DNA molecule containing a fluorescent bead at the upstream end. The rate of movement and progression of the polymerase can be followed by observing the position of the bead over time using a fluorescence microscope. In this protocol, the attached polymerase is transcribing a DNA molecule with a head bead at its downstream end. So this is an experimental procedure to view this transcription process. Then, even though polymerases are relatively powerful motors, these enzymes do not necessarily move in a steady, continuous fashion, but may pause at certain locations along the template or even backtrack before resuming their forward progress. A number of elongation factors have been identified that enhance the enzyme's ability to traverse these various roadblocks. In some cases, as occurs following the rare incorporation of an incorrect nucleotide, a stalled polymerase must backtrack and then digest away the 3' end of newly synthesized transcript and resynthesize the missing portion before continuing its movement. The ability of RNA polymerase to recognize and remove a misincorporated nucleotide is referred to as proofreading. This corrective function is carried out by the same active site within the enzyme that is responsible for nucleotide incorporation. At this point in this discussion, it is useful to examine the differences in the process of transcription between bacterial and eukaryotic cells. Next topic, transcription in bacteria. Bacteria such as E. coli contain a single type of RNA polymerase composed of 5 subunits that are tightly associated to form a core enzyme. If the core enzyme is purified from bacterial cells and added to a solution of bacterial DNA molecule and ribonucleoside triphosphate, the enzyme binds to the DNA and synthesizes RNA. The RNA molecule produced by a purified polymerase, however, are not the same as those found within cells because the core enzyme has attached to random sites in the DNA, sites that it would normally have ignored in vivo. So inside of bacterial cell. If, however, a purified accessory polypeptide called sigma factor is added to the DNA RNA polymerase before it attaches to DNA, transcription begins at selected locations. Attachment of sigma factor to the core enzyme increases the enzyme's affinity for promoter sites in DNA and decreases its affinity for DNA in general. As a result, the complete enzyme is thought to slide freely along the DNA until it recognizes and binds to a suitable promoter region. The next crystallographic analysis of the bacterial RNA polymerase reveals a molecule shaped like a crab claw with a pair of mobile pincers or jaws enclosing a positively charged internal channel. As the sigma factor interacts with the promoter, the jaws of the enzyme grip the downstream DNA duplex which resides within the channel. The enzyme then separates or melts the two DNA strands in the region surrounding the start site. Then, strand separation makes the template strand accessible to the enzyme's active site, which resides at the back wall of the channel. Then, initiation of transcription appears to be difficult undertaking because an RNA polymerase typically makes several unsuccessful attempts to assemble an RNA transcript. Once about 10 nucleotides have been successfully incorporated into a growing transcript, the enzyme undergoes a major change in conformation and is transformed into a transcriptional elongation complex that can move progressively along the DNA. In the model, the formation of an elongation complex is followed by release of sigma factor. So, as noted above, promoters are the sites in DNA that bind RNA polymerase. Bacterial promoters are located in the region of RNA strand just preceding the initiation site of RNA synthesis. The nucleotide at which transcription is initiated is decoded as +1 and the preceding nucleotide as -1. Those portions of the DNA preceding the initiation site toward the 3' end of the template are said to be upstream from that site. Those portions of the DNA succeeding it towards the 5' end of the template are said to be downstream from that site. So, analysis of DNA sequences just upstream from a large number of bacterial genes reveals that two short stretches of DNA are similar from one gene to another. One of these stretches is centered at approximately 35 base upstream from the initiation site and typically occurs as the sequence TTGACA. This TTGACA sequence, known as 35 element, is called a consensus sequence, which indicates that it is the most common version of a conserved sequence, but that some variation occurs from one gene to another. The second conserved sequence is found approximately 10 base upstream from the initiation site and occurs at the consensus sequence TATA80. The site in the promoter, named the Pribnow box after its discoverer, is responsible for identifying the precise nucleotide at which transcription begins. As the sigma factor recognizes the Pribnow box, amino acid residues within the protein interact with each of the 6 nucleotides of the TAT80 sequence of the known template strand. 2 of these nucleotides are flipped out of the nucleotide stack and into the core of a protein. This action likely initiates melting of the adjoining region of promoter DNA and formation of the transcription bubble. And bacterial cells possess a variety of different sigma factors that recognize different versions of the promoter sequence. The sigma 70 is known as the housekeeping sigma factor because it initiates transcription of most genes. Alternative sigma factors initiate transcription of a small number of specific genes that participate in a common response. For example, when E. coli cells are subjected to a random rise in temperature, a new sigma factor is synthesized that recognizes a different promoter sequence that leads to the coordinated transcription of a battery of heat shock genes. The products of these genes protect the proteins of cell from thermal damage. Just as transcription is initiated at specific points in the chromosome, it also terminates when a specific nucleotide sequence is reached. In roughly half of the cases, a ring-shaped protein called Rho is required for termination of bacterial transcription. Rho encircles the newly synthesized RNA and moves along the strand in a 5' to 3' direction to the polymerase, where it separates the RNA transcript from the DNA to which it is bound. In other cases, the polymerase stops transcription when it reaches a terminator sequence. Terminator sequence typically folds into a hairpin loop that causes the RNA polymerase to release the completed RNA chain without requiring additional factors. Then, transcription and RNA processing in eukaryotic cells. As discovered in 1969 by Robert Rhoda in the University of Washington, eukaryotic cells have 3 distinct transcribing enzymes in their cell nuclei. Each of these enzymes is responsible for synthesizing a different group of RNA. Plants have 2 additional RNA polymerases that are not essential for life. No prokaryote has been found with multiple RNA polymerases, whereas the simplest eukaryotes, yeast, have the same 3 nucleotypes that are present in mammalian cells. This difference in number of RNA polymerase is another sharp distinction between prokaryotic and eukaryotic cells. Surface structures of RNA polymerase from each of the 3 domains of life like archaea, bacteria and eukaryotes are different. Several features are apparent from a close examination of these 3 structures. It is evident that the archaeal and eukaryotic enzymes are more similar in structure to one another than are the bacterial and archaeal enzymes. This feature reflects the evolutionary relationships between archaea and bacteria, respectively. The subunits that make up each of the proteins depicted are indicated by a different color, and it is immediately evident that RNA polymerases are multi-subunit enzymes. Those subunits between the different enzymes that are homologous to one another are derived from a common ancestral polypeptide. Thus, it is also evident that the RNA polymerases from the 3 domains share a number of subunits. Although the yeast enzyme has total of 12 subunits, 7 more than its bacterial counterpart, the fundamental core structure of the 2 enzymes is virtually identical. This evolutionary conservation in RNA polymerase structure is very important, and that shows homologous regions of the bacterial and eukaryotic enzyme at much higher resolution. So our understanding of transcription in eukaryotes was greatly advanced with the 2001 publication of the X-ray crystallographic structure of yeast RNA polymerase II by Roger Kornberg and colleagues at Stanford University. As a result of these studies and those of other laboratories in subsequent years, we now know a great deal about the mechanism of action of RNA polymerases as they move along the DNA, transcribing a complementary strand of RNA. A major distinction between transcription in prokaryotes and eukaryotes is requirement in prokaryotes for a large variety of accessory proteins or transcription factors. These proteins play a role in virtually every aspect of the transcription process, from the binding of the polymerase to the DNA template, to the initiation of transcription, to its elongation and termination. Although transcription factors are crucial for the operation of all 3 types of eukaryotic RNA polymerase, they will only be discussed in regard to the synthesis of mRNA by RNA polymerase II. All the major types of eukaryotic RNA like mRNAs, rRNA and tRNA are derived from precursor RNA molecules that are considerably longer than the final RNA product. The initial precursor RNA is equivalent in length to the full length of DNA transcribed and is called the primary transcript or pre-RNA. The corresponding segment of DNA from which a primary transcript is transcribed is called transcription unit. Primary transcripts do not exist within the cell as naked RNA but become associated with proteins even as they are synthesized. Primary transcripts typically have a fleeing existence, being processed into smaller functional RNAs by a series of cut and paste reactions. So, RNA processing requires a variety of small RNAs and their associated proteins. In the following sections,